Hello everyone. I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.G. Hariharan. So today we are going to discuss about one of the technique of enumeration of bacteria, that is by using a flow cytometer how we can perform a total count of a given sample of bacteria. So this lecture I have divided into two parts. So first we will get a brief outline about the different methods available for the enumeration of a bacteria, and in the second part we will elaborately discuss about the flow cytometer. So what is the basic components present in a flow cytometer? So what is the basic principle underlying how it measures the cell count, right? So first we will understand the measurement of microbial growth. So the core objective of enumeration of bacteria or enumeration of microorganism is based on the measurement of microbial growth. So the microorganism growth is very critical part, especially for performing and fermentation production process. So in a fermentation process such as production of antibiotics or vitamins, the important thing is in what stage the production cell, what stage the microbial cell is present, that is the microbial growth population should be measured. So the measurement of microbial growth is done for the determination of one, growth rate, and second is the generation time. So these things is critical for the product formation. So I have elaborately given a separate video lecture about the bacterial growth curve and the mathematics about the calculation of this growth rate and the generation times. So you can view in this aspect. So to measure this, we need a techniques to count the bacteria in a given sample. So for example, I have taken a test tube of a sterile broth media. So I have taken an organism, a loop full of organism is added. At the 0 minute, what is the total count of the cell or what is the viable count of the cell? We have to calculate it. And after a specific interval, we have to count how much cell is present in that particular sample. So that is considered an enumeration of bacteria or measurement of microbial growth. There are different methods are available for this measurement purpose. So the measurement can be based on the cell count, or the measurement can be based on the cell mass, or the measurement can be based on the cell activity. So whatever the counting method is there, so the total objective is what is the population of the cell present in that particular sample. So these techniques were further classified into the following methods. So the first one, that is the measurement based on the cell count. So this is divided into two types. One is the total count, which is a direct method. We can count either by a microscopic method or by using a electronic particle counter. So one of the method of electronic particle counter today we are going to discuss is the flow cytometer. So the microscopic methods are available like Breed's method, Wright's method, Petro-Forstner chamber has been used for this technique recently. So all these things will give an idea about the total count. So we can directly count how much cells is present in a particular sample. Whether the cell is living or dead doesn't care about it. We will calculate only the total count, total cell. The second method of this cell counting is the viable count. So the viable means the living cells. The living cells, how much it is there in a particular sample can be calculated by this method. It is an indirect way of counting the cell. So why we call it as an indirect way is that the living cell only has an ability to divide, whereas the dead cell doesn't have an ability to divide. The living cells divide once it is kept in an appropriate medium. It will divide and forms colonies. So the number of colonies will be equal to the number of cells. That was the basic indirect objective of counting the cell. That's why it is considered as an indirect method. There are two techniques available in this. One is the plate count method, and second is the membrane filtration method or membrane filter count method. So both the things, the objective is to count number of viable cells. And the second major technique is the measurement of cell mass. So population of the cell can be determined in terms of its weight. That is called as a cell mass. There are two techniques available. One is a direct method and second is an indirect method. In direct method, one method we can directly weigh the cell. That is the, we call as direct weighing of dry weight of the cell, which is most popularly used. And second is the measurement of cell nitrogen, which also gives the number of biomasses present in a particular sample. So whereas in industries, the indirect method is most widely used with the help of either a spectrophotometer or with the help of a nephelometer. So both the things will measure the cell mass based on the formation of the turbidity. So this gives an indirect way of calculating the cell mass. And the third one is less used, which is called as a cell activity method. It's an indirect method. It is based on the biochemical activity of the cell population. Based on the cell volume can be measured. So this is the common method of enumeration of an microbial cells, or most commonly we will consider as an enumeration of the bacterial cell. So in this thing, so we will discuss about the first one, that is the cell count method by total count. So the word total count tells that it includes both viable cells, that is the living cells, and the dead cells. So the total count comprises of both the things. Whereas the viable count will only calculate the living cells. So the total count is a direct counting technique, because we will directly count how many cells are present in a particular sample. So this is the most satisfactory method of counting the cells. So it can be done by two ways. I have already told, either by a microscopic and optical method, or by an electrical conductivity method using an electrical counter. So you can see there are different types of electronic counters are available for this total cell count. So there are two widely used equipment, is the Coulter counter, and second is the flow cytometer. So I have given a separate video lecture on the Coulter counter. So in this lecture we will discuss about the flow cytometer. So let us understand about the flow cytometer. So the name itself tells us, so if a liquid flows, based on the flow, the cyto means the cell is calculated. The meter will measure how much cells is present. So the flow cytometer is the one of the good machine currently available for analyzing number of cells. So whether the blood cells should be counted, or where microbial cell present in a sample can be counted, or particles present in a suspension can be counted, or in an aerosol, whatever things, the flow cytometer has been wide application. So if you see that it is, since it is having a wide application, not only it has been widely used, but also it is a powerful tool. So because why we call the flow cytometer is a powerful tool is that it can do multiple parametric analysis at a single time at a very fast rate. That means it can perform the physical as well as chemical characteristics of the particular particle or particular cell at a very fast rate. That, for example, you can measure the things about 1,000 of particles in a second itself. That's why it is a very good powerful tool for analyzing the particle as well as cell, both its morphological and chemical characteristics. So it is a rapid and quantitative methods of analysis. Apart from analysis, it also helps in the purification of the cell also from the suspension. So the flow cytometer has its wide application. So that's why we will understand the basic nature, how the flow cytometer works. So if you see the components present in the flow cytometer, we can be broadly divided into three parts. One is the fluidic part, second is the optic part, and third is the electronic part. So every system components works in a particular fashion to give a final output what we want to know. So if you see the fluidic system, so in fluidic system, what is that? Is a simple system. It carries a cell called as a flow cell. Into that, the sample has been added or injected. So after adding this sample, what happened is that the flow cell carries a sheath of fluid. So it helps into aligning this sample, which is carrying the microbial cells or particles, into a single layer. So the sheath fluid, what makes is that it makes the sample fluid to pass through the narrow channel, each cell individually through this path. So why it has to pass individually is that through this narrow channel only the light beam is passed through it. So the flow seal is an fluid dynamic system. So this fluid dynamic system, otherwise I can also call it a hydrodynamic system, because it makes a cell to pass one cell at a time through this narrow channel for analysis purpose. So this you can see from this picture. So this is the fluidic system. So this is the flow cell. We have added the sample. It carries different cells. If it's a blood cell, it contains monocytes, leukocytes, or erythrocytic cells, whatever cells it may be. So what happened is that this fluidic system, because of its hydrodynamic nature, it makes to flow the single cell at a time through this narrow channel. Right. So this is the basic objective of the fluidic system. If the fluidic system fails to pass the single cell at a time, what happens is that it gives to the confusion of the counting of the cell as well as the physical characteristics of the cell. So the second part is an optic system. So the optic system is the core system. So the fluidic system, once sends a single cell, this optic system comprises of three things. One, it carries a light source which may be ranged from a UV light up to a far IR. That means the generally the EMR spectrum of an light source, if you see that it starts from cosmic waves to the radio waves, but for our analytical purpose it will be present between the ultraviolet region to the far IR region. So the light source will pass the lights through this, and it carries various filters. The filters will capture how much light is emitted from the each particle or lights. So finally a detector like photomultiplier tube or photodiodes will be present, which helps in the amplification of the received light source, right? So the major one is that the light source. Once the light source pass through this, it has to pass a single wavelength at a time. So single wavelength at a time, it is passed from the entire region to that particular flow through cell. Then it is collected by the filter and it is processed by the detector. So this light pass through the flow through cell. In the flow through cell, the particles or the cells will be present, that it pass through the cell and it is collected in the filter. So how much intensity of light we have passed, how much it is observed, how much it is collected, or how much it is emitted, like that it will makes the filter will collect the light source, then it will process through the photomultiplier tube. So I have told that the light source or from UV to far IR region is commonly used. So in this picture you can see the light source is passed, the lens will send a particular wavelength of light, then the filter will collect. So the optic system like this, different filters will be present. Whenever it pass, it will be collected by the photomultiplier tube, which acts as a detector, and it is amplified with the help of the electronic system. So the electronic system helps to detect into give a graphical representation what the data it has been collected from the detectors. So this I have put in this picture. You can see that the entire three system, how it works. The fluidic system will helps to align the sample into a single cell or single particle to pass through this flow cell. So then the optic systems comes into play. It sends the light source as a laser beam which hits through this flow cell and it gets collected with the detector. So it carries different filters. The different filters will absorb light at different ranges, and with the help of photomultiplier tube or photodiodes, it gets collected by the electronic system. So it gets different intensity of light, then it is digitally converted by the electronic system, then we get a graph. So in the graph we get both the characteristics. If there are different types of cell, what is the size of the cell, we can measure it. How many cells there, we can measure it. If it is a particle like a suspension, we can measure the particle surface morphology, particle diameter, even though we can calculate with this flow cytometer. So I think this lecture will be very helpful for you understanding about the flow cytometer in a brief manner. So you continuously watch my future lectures in this microbiology, or otherwise I have more lectures in the biotechnology and immunology also. You can view through my channel. So thank you very much. Thank you.